Hello students, welcome to the EPG Pathshala. I am Dr. Rajeshwari Sinha and in today's lecture we are going to learn about the secondary structure of proteins. We know that there are four major levels of protein structure, namely primary structure, secondary structure, tertiary structure and quaternary structure. In this lecture today, we will take a deeper look at the secondary structure of proteins. Let us first understand the objectives of this lecture. We will understand the basis of formation of secondary structure, the stability factors of the secondary structure, explain the various types of secondary structures found in proteins, and also look at the significance of each type of secondary structures. We hope you have an enjoyable time listening to this lecture. In this module, we have a look at the concept map that we are going to follow. We see how the primary structure of protein moves into the secondary structure and then into tertiary and quaternary structures. We will look at each of these in detail in this module. Introduction. Proteins must fold in such a way that it is stable, flexible, able to work in the correct site in the cell and capable of being degraded by cellular enzymes. Secondary structures consist of local regions of polypeptide chains formed into structures generally stabilized by hydrogen bonds. Several years before the elucidation of the first complete protein structure, Pauling and Corey predicted the existence of these secondary structures in 1951 with the help of fundamental chemical principles and experimental observations. The discussion of secondary structure generally covers common regular folding patterns of the polypeptide backbone. The stable and most widely occurring secondary structure of proteins are the alpha helix and the beta sheet conformations. Alpha helices are right-handed helices with 3.6 amino acid residues per turn. In these structures, hydrogen bonds are formed parallel to the helix axis. Beta sheets are parallel or anti-parallel arrangements of the polypeptide chain in which hydrogen bonds are formed between two or more polypeptide strands. Stability of protein secondary structure. The secondary structure exists to provide a way to form hydrogen bonds in the interior of a protein. These hydrogen bonds are just replacing those originally made with water. As the protein folds, a number of hydrogen bonds to water get broken. If these hydrogen bonds are replaced by hydrogen bonds within the protein, there is no net change in the number of hydrogen bonds. The figure below shows the solvation process in protein folding. Let us look at the secondary structure stabilization. On the left, we are looking at the side of a beta sheet in which every other residue is on the same face of the sheet. On the right hand side, on looking down the axis of an alpha helix, we observe that the residue sequence is numbered. The angle between the residues is 360 by 3.6 residues or about 100 degrees. Alpha helix. The alpha helix possesses a rigid conformation that maximizes hydrogen bonding while staying within the allowed rotation angles of the polypeptide backbone. The backbone of peptides in alpha helix is formed by strong hydrogen bonds between each carbonyl atom and the amide hydrogen of an amido acid residue located 4 residues further down the chain. It is a common secondary structural element of globular proteins, membrane spanning domains and DNA binding domains. The core of the helix is tightly packed, that is, its atoms are in van der Waals contact across the helix, thus maximizing association energies between atoms. The trans side of the amino acids project backward and outward from the helix, consequently avoiding steric hindrance with the polypeptide backbone and with each other. For a polypeptide made from L-alpha amino acid residues, the alpha helix is right-handed with torsional angles phi of minus 57 degrees and psi of minus 47 degrees, n equals to 3.6 residues per turn and a pitch of 5.4 angstrom. In this slide, we have a look at how a right-handed alpha helix looks like. Students, we hope you are finding this lecture useful. We have now come halfway through this lecture. As we see that the secondary structure of proteins is primary alpha helical structure and the beta pleated sheath structure. We saw the alpha helix structure in detail. An important thing to remember in this case is that in the alpha helical structure, intermolecular hydrogen bonds do not predominate. Instead, it is intramolecular hydrogen bonds that predominate, and the helix is stabilized by such intramolecular hydrogen bonds within the molecule. We now move to the next section and see how the beta pleated sheath structure is different from the alpha helical structure. We will also see some other types of secondary structures which are non-repetitive secondary structures such as bends, loops and turns. Let us take a look. We will now look at the beta sheaths. In beta conformation, hydrogen bonding occurs between neighboring polypeptide chains rather than within one as in case of alpha helix. The beta conformation is a more extended type of conformation of polypeptide chains whose structure has been confirmed by the X-ray analysis. It is characterized by a zigzag pattern of the polypeptide backbone rather than helical. The zigzag polypeptide chains can be arranged side by side to form a structure resembling a series of pleats termed as beta pleated sheaths. The beta pleated sheaths are of two types: anti-parallel beta pleated sheaths and parallel beta pleated sheaths. In the anti-parallel beta pleated sheaths, neighboring hydrogen bonded polypeptide chains run in opposite directions. On the other hand, in parallel beta pleated sheaths, hydrogen bonded chains extend in the same direction. The R groups of adjacent amino acids protrude from the zigzag structure in opposite directions, giving rise to an alternating pattern. The repeat period is 6.5 angstrom for the parallel conformation and 7 angstrom for anti-parallel conformation. Here we look at pictorial diagram of the anti-parallel and parallel forms of the beta pleated sheaths. Non-repetitive secondary structure. Bends, loops and turns are non-regular structures that do not contain a repeating element. These are secondary in nature. They are characterized by an abrupt change of direction and are often found on the protein surface. These non-regular structures are usually more flexible than the relatively rigid helices and beta pleated sheaths. They often form hinge regions that allow segments of the polypeptide chain to move as a compound binds or to move as the protein folds around another molecule. Beta turns refer to short regions generally involving 4 successive amino acids. They connect strands of anti-parallel beta sheaths resulting in tight 180-degree turn. Proline and glycine are often found in beta turns. In this slide, we look at the non-repetitive secondary structure examples: a helix loop helix motif and bends, loops and turns, which are non-regular secondary structures that are involved in stabilization of the 3D structure of the proteins. Another form of turns occurs less frequently, such as gamma turns, which is a 3 residue turn with a hydrogen bond between the first and the third residues. The polypeptide segments connecting the helices and the beta sheaths are said to have a coil or loop conformation. Loops serve a number of key biological roles, such as for many enzymes, the loops that bridge domains are responsible for binding substrates often containing amino acyl residues that participate in catalysis. While loops lack apparent structural regularity, they exist in a specific conformation stabilized through hydrogen bonding, salt bridges and hydrophobic interactions with other portions of the protein. Prediction of secondary structure. Prediction of secondary structure can be done or can be made possible through various bioinformatic tools or software. Some online softwares are as follows: APSSP, which stands for Advanced Protein Secondary Structure Prediction Server; CFSSP, which stands for Chow-Fasman Secondary Structure Prediction Server. YASPIN is an HNN hidden neural network secondary structure prediction program that uses the PSI-BLAST algorithm to produce a PSSM for the input sequence, which it then uses to perform its prediction. The PSI-PRED protein sequence analysis workbench, and lastly the NPS network protein sequence analysis. Students, we have now come to the end of this lecture. Thanks for being there with us. We will now see a brief summary of what we have learnt in this lecture today. Secondary structures are the regular arrangement of amino acid residues in a segment of polypeptide chain in which each residue is spatially related to its neighbors in the same way. The most common secondary structure of proteins are the alpha helix, the beta conformation and the beta turns. The secondary structure of the polypeptide can be totally defined if the phi and the psi angles are known for all amino acid residues in that given segment. Thanks for watching this lecture on the secondary structure of proteins.